To the cloud. Okay, we have Sally, Hesong, Liam, Yujong. Okay, can we start with Hesong? I know this is gonna be your last lab meeting on site.
Oh, I'm starting? 잠시만요.
Good morning.
Morning. 잠시만요. I'll start with darts. Let me share my screen. Okay, so for NEOC, it was submitted to CCR last Friday. EAC submitted to Therapeutic Advances in Med-Onc, still pending decision. Thyroid, for thyroid, I wonder if you got like a final feedback from Dr. Kurzrock?
Let me check. But move on. I'll check.
Okay, and then clear cell gyne, I prepared all the manuscript and figures and gave it to Liam. Liam, can you update us on like your timeline for when you're planning to submit this?
Yeah, so this one I plan to submit today or tomorrow.
Okay, thank you. For desmoid, it was submitted to JITC last Friday. Vulvar and small cell ovarian, we're still collecting COIs, and these are due on March 8th. I'll be in Korea back then. All right, at this time. And then for sarcomatoid, I finished my first draft. Would you like to take a look right now? Or
Yeah.
should I? Okay.
Yeah, let's look at it.
Yeah, sorry, this took a long time because all the data were so scattered. It was pretty challenging, but I finished this like this morning. Here. Okay. Oh wait, sorry, one sec. Let me turn off scripting. Also, Stas is supposed to join today to give you an update on the early stage non-small cell lung cancer low TMB heterogeneity project. We finished revising everything.
Good.
Yeah. Are you seeing this screen? Sorry, okay. For keywords, I thought of including both, like pulmonary sarcomatoid and sarcomatoid of the lung. These
That's
two
fine.
terms are used interchangeably. Okay.
Yes.
Besides that, maybe starting with abstract. So by the way, I've asked Megan to update the toxicity table because the version I have has a total number of 13 patients, whereas 14 patients were evaluated. So I'm trying to confirm why there's that discrepancy.
I've seen discrepancies happening from sometimes eligibility criteria when to be eligible for a response assessment and to be eligible for toxicity, you have to be on drug for a certain amount of time.
I
Sometimes
see.
that they have that kind of definition, but But I think for this paper, it should be same in my mind, but ask her, she'll know.
Maybe this one got omitted because it was too short,
Maybe.
something like that. Okay.
Yeah,
I'll
maybe.
just confirm.
And yeah, and you can explain that in the method that, one patient was not eligible for toxicity assessment, something like that. Just add that in the method. If it is truly the case.
Yeah. Just to give you a rough timeline. So if Megan gets back to me today, then I think I can revise it by the end of the day. But tomorrow until Sunday, I'll be in New York, and then Tuesday I'm going back to Korea.
Okay.
So if she doesn't get back to me today, then it might be delayed for a few days. But I'll try to see if I can make time.
Okay. Okay, understood. Okay, so can you show me?
Yeah, it's good. Okay. Introduction, I've made some like minor edits, so if you could go over it again.
Okay. Oh, so did you look at the MET
Yeah.
mutation frequency?
I
It just says frequency. Does it have any number? No?
I wonder why.
Or is that in discussion? Maybe, yeah, because we talked
Yeah, I'm just trying to remember. Oh yeah, I think
33.
I wonder why I took that. So this was just one study.
Mm-hmm.
Maybe this one, I wonder why I took that out, like why I removed it. But so you want me to put the exact like stats, right?
No.
Oh,
For the introduction, it's fine.
okay.
For, yeah, discussion,
Oh, here. That's the one.
is this introduction? Okay.
Yeah, this is introduction. Yeah.
Well, you have one citation, number 8.
Yeah, the thing is like JTO only allows 50 references. It's at max right now, it's 50, and this is like a review paper that goes over like the molecular landscape of this cancer. If I have to cite it, then this includes like more than 10.
10 other paper because it's a review paper. Okay.
Right.
So yeah, it's a big range. Wow. KRAS, EGFR, MET. But MET exon can be really high as 31%. Wow. Okay, I think that's enough. Yeah. Yeah, it's unclear, but there's a potential. Okay. Disease-free survival was better. Do you have response rate for MET TKIs in the discussion, or is it just the PFS?
Yeah, I just have what I found in each study.
So, what I'm a little bit confused is if you have the use of MET TKI targeting MET exon 14 mutation, wild about and
Oh, wait. Uh,
like the sentence,
It's-
like, yeah.
It's the mutations that's associated with reduced disease-free survival. That's what I found in the one of the papers. Huh.
No, I
Okay.
think, first of all, this sentence is too long. It just break it down into, idea, value. So
Hmm.
use of TKI, is it the use of TKI?
Okay, yeah.
The mutation
Oh,
itself?
No, I don't remember.
And
Okay.
then
Probably like TKI.
disease-free survival is pertinent to limited stage or local stage because you're disease-free. You know what I mean? After surgery. If you're thinking of advanced stage, you're talking about progression-free survival
Mhm.
with the use of med TKI. So this is for everyone in our lab meeting. So I'll give you an example. HER2 mutation itself in breast cancer is what? Poor prognostic factor, right? But if you give those patients HER2 targeted therapy, then HER2 becomes what? Favorable prognostic factor because they have additional good targeted therapy option. So here in sarcomatoid lung cancer, MET mutation itself without treatment, is that a good thing or a bad thing? Now for stage 4 sarcomatoid, I think it is likely to be a good thing because you have additional treatment option, which is the MET TKI. So we have to make that distinction clear.
Yeah, I'll take a look. Yeah, I'll take a look and get back to you.
Okay. So separate those limited stage, advanced stage, separate effect of the mutation itself, and separate the effect of the TKI. So those are all different things.
Do you want me to focus on the advanced stages because that's where the treatment options are lacking? So find like the PFS OS.
Yeah.
Okay. Oh, cool.
But for TKI, response rate is also a outcome of interest.
Mhm.
In a simple term, if you give the drug, will the tumor shrink or not? It's a very simple answer. But my concern is because of the rare population, small n, but those data hard to find, scarce. Okay. Uh, okay, okay, looks good.
I just need to update the methods and results section.
Okay. So this is the only one. No one did Nivo EP for sarcomatoid lung, right?
Right, but there was this another study that did durvalumab.
Durvalumab, yeah. What was the grade 5 AE? The cause?
This one?
Yeah.
I need to review. Well, again, like I need the updated toxicity. Well, I'll take a look at the version I have right now.
Yeah. If it's unclear, ask Megan. So the reviewer is gonna ask, like, what was the cause of the mortality? And sometimes,
Oh, it
you
says
know,
respiratory failure.
mm. And the readers will know that this respiratory failure may be, you know,
Mm.
so for all of you, if you do clinical trial, adverse event happens, you have to report it. And that's regardless of whether this is drug induced or not. And sometimes you don't know for sure. So that's where possible, plausible, most likely, or presumed, there's different categories you check as an investigator. Whatever you check, if it's possible or cannot rule out, it gets reported as a drug AE. But we all know it could be from pneumonia, infection, PE. Sometimes if they die, you never know what the exact cause was. Um, so we don't know. The answers we don't know.
Dr. Kurzrock confirmed for us, at least for other cohorts, that the ones reported in the toxicity table are all drug related
Yes,
adverse events.
yes.
Yeah.
Okay. So compared to durvatremi, how is our study different?
That's what I was trying to show. Like in terms of OOR, PFSOS, it's pretty comparable, but durvaltremi resulted in higher frequency of adverse events.
Hmm. Hmm. Comparable clinical. Okay. 26%. What do we have our duration of response?
That's PFS. Median PFS was 6.1. Here it was 5.9.
Hmm.
OS 10.9.
Can
Yeah.
we ask Megan to give us duration of response?
Mm-hmm.
Do we yeah, so that we can report and kind of compare with the study. How many PRs do we have?
1 CR and 3 PRs,
Okay.
and 3 SC durable, greater than 6 months.
Hmm. So duration of response is counted only among PR and CR,
Mhm.
not this. So yeah. So let's get that. So we can add that to our abstract as well as result, and also use that to say compare it with the durvatremi result. Okay.
But would you still say it's like comparable clinical response?
Yeah.
Okay.
I hope our duration response may be longer
Mhm.
than 10 point. Can you see if it's for
For
the
P.
PRs,
Oh,
for the
wait.
PRCRs?
We have, sorry, this might be for another course. Let me pull up the PDF I got. Oh, here, sorry about that. That was not updated.
That wasn't our commentary?
No. This is it, sorry.
Oh, yeah. So because this one has CR, okay.
Mhm.
So look, apart from one's PR,
Right,
all three of them are past 20 months,
yeah.
right? So if
Mhm.
you,
Over 2 years.
yeah, so our duration of response seems to be much better than 10,
Mm.
right?
Mhm.
Yeah. So it's not only you can say comparable response rate and PFS, but the duration of response for the ones who responded maybe double uh the duration of response of that of durvaltremi. You know,
Yeah.
durvaltremi is usually given with the tremi finishing after five dose. Nivo EP is EP continued forever.
Mm.
So you can discuss that, go back to that paper and look at the method, how tremi is given. But all the durvaltremi trials that I know so far had tremi given only 5 times or 4 times. And then only durva gets continued. Maybe that's a difference that might have led to difference in duration of response. So you're welcome to discuss that in the discussion section.
So for Nivo, it'd be
Nivo
those combinations continued
forever.
or is it just okay?
They continue forever versus tremi usually is given limited time.
Mhm.
But go back to that paper and find out if that's actually what was done. And for the readers, when you say better duration of response, you can actually put number month, this month with Nivo AP versus the, for the just the readers' convenience. Do you know in Durva Tremi study, they looked at PD-L1 expression level?
Um, no.
Oh, go back up. 옥찬영, 김범석, 김미소.
Oh yeah.
This is the same group in Seoul National University. This
Yeah.
is 2020. So this
20.
study, 허대석. Uh, so this is while he was still a faculty at Seoul National University, 옥찬영 선생님.
Mm.
And correspondence is 김범석, my friend. That's so funny. Yeah. Can you look at PDL1?
Yeah. Again, they didn't look into the
They
expression.
didn't report? Oh.
No. this isn't published. Oh. Okay, I guess. Hi. Yeah, sorry, it didn't search when I initially looked it up. So 14 patients out of 18.
Hmm. Did they look outcome based on PDL1? Can you see if they did?
Here it says it was not correlated.
Oh. Yeah, look at this. So it would have been great if we had, mutation and all that, then we could create graph like this. I think it's a shame that we didn't do this. We have the sequencing, but we just didn't do we just couldn't get to this cohort anyway. So do you think PDL1 high has more responders?
So at least for the lower than 50%, it was 2 progressive disease, not
Mm.
assessed, and one partial.
Uh,
Or sorry, this is stable.
mm,
So the yellow ones are less than 50, and then the red ones are greater than 50.
mm. Okay. So maybe some trend, maybe.
Like 2 partial responses among the high PD-L1 group.
Right. One is unknown. Yeah. So in our core, do we have PDL1 expression information?
No, not with circumtoid.
No, no.
No. Should I ask Megan though? Because what I was
Yeah,
given, I could not see anything.
because I know, right. I think PDL1 is part of the CMAC as well. So someone like Kara has to run it. So we don't have it.
Mm.
It's okay. Well, sometimes, well, I think it wouldn't hurt to ask. Just ask Megan, do you have any recorded PDL1 expression level anywhere for this cohort? Just ask. I think 90% its answers no, but sometimes the individual site will record PDL1 from their own pathology report, right? Because it's lung cancer, lung cancer, every institution does it. Okay, I'm gonna ask this to Liam. Liam, when you were and then maybe Haesung, you looked at the path reports too. In the path report, because this is lung cancer, did you see PDL1 reported in any of the path report?
I don't know if they was reported in all of the patients, but I do think like it's been a while since I looked at them, so I'm not exactly sure, but I think a few of them had mention of PDL1 levels, but not all. That's what I recollect now, but I can go back and check.
Yeah, because we can't go back and ask the sites for that, but let's say how many total in our cohort, Haesung?
14.
14. I think it doesn't make any sense to report. Maybe let's say we have 3 patients with PDL1 information. Like, what are we gonna make sense out of it? So, if I don't know, maybe 30%, um, something like that. If you can just skim through the path report and at least 30% has PDL1, you can report it. And do similar things as this paper have done. Among the high expressors, lower expressors, how many had response or not, things like that you can describe. Um, and not available, just if it's not there, just say not available. But it's less than 30, I feel like we don't have to report it.
Another thing I'm concerned is that like every site has different assays for PD-L1 and they might have a different cutoff for what's high and low. So it could be difficult to analyze the response rate among high versus low.
Mm-hmm. It is, but that's what's being done real world. We use different assays, but the high cutoff is always 50. And yes or no cutoff is always less than 1% or greater than 1%. So we know every essay is different, but still, we think they are still within that category of comparable essays. Yeah.
So if we do have enough PD-L1 expression level, then mention it here and also mention the PD-L1 expression level of this cohort. But if not enough PD-L1 expression levels, then don't mention it.
Correct. And if
Even for this cohort, right?
yeah, even for that cohort, don't mention it. But if we have like 30% or above, mention it in the method, in our result, in our discussion. Yeah. Because I think because this is lung cancer, the more I think about it, the reviewer is gonna ask. Reviewers are going to ask, yeah.
Okay, so 5 or more patients with this data, then mention in both cohorts.
Yeah. Okay, next. So the next paragraph, the use the lower paragraph, inclusion of immunotherapy. Can you at least say what those immunotherapies are?
Yeah, I mentioned it here.
Okay.
So this is
Same paper? Okay.
it's the same paper, yeah.
Okay. Is this a Chinese paper?
There were many Chinese papers. Yeah.
Mm.
Yeah.
'Cause those immunotherapies are approved in China and never used in United States. Isn't it interesting? There's a lot of Chinese yeah,
This one, Tislet and the Sintil, right?
right.
Camera. Oh, camera too, yeah.
Yeah,
Mhm.
a lot of people are using it there. But anyway, what worked best?
Among these?
It's all combined, so we don't know, right? It's all combined.
Right. Yeah, they didn't do a subgroup.
Mm. Okay. Okay, as a monotherapy, response rate is what was it? Go up. Can you go up? Monotherapy. Well, what's the monotherapy response rate? 57%
I'm
is
not.
this monotherapy or combined?
So this one is combined. The 3 patients received monotherapy and the rest 18 received combo. So
Oh,
you're looking for a
okay,
subgroup, right?
so that's all
What?
combined, all combined.
Yeah, I don't
No, well, I don't
recall
have to know the
finding.
three patient, it's okay, but do you
Yeah,
have a monotherapy response rate?
I think the one that after this is recent study.
That's a combo.
This is combo
Combo.
as well. Yeah, that's it
So
for
there,
first line options.
right, but there's no mono first line or second line?
Second line, there were a lot of like case reports. So these ones are monotherapy as second line option. Responses exclusively in high expressors. Um, yeah, 50.
But do you have combined or just for high? Oh,
So no responses in low PD-L1 in this cohort at least.
oh, exclusively, okay.
Exclusively, yeah.
Oh, okay.
So 41 patients had high, and among them 24 had responses.
Mm. It would be good to have among everyone, you know?
Well, then it will be 24 out of 49.
49, so just 8 had low, low expression or
Looks
negative.
like it, yeah.
Hmm.
Yeah, 8 with low negative unknown.
Hmm. A low negative unknown, okay. So that's a pretty good response rate uh, for
Sounds like,
monotherapy.
yeah.
Okay. So I would write among all second line mono, this is the response rate, and then mention among high expressors what so just mention the objective response rate in second line in general, and then go to the high expression level subgroup so that people know.
Mhm. And then in this cohort, regardless of PD-L1 expression levels, these monotherapies using as second line resulted in 40.5% response rate.
Hmm. Yeah. So this is pretty good. This is pretty good because in not well in non people would be interested to know what what's non-sarcomatoid data, but that usually the second line ORR is a lot lower than that. Okay. And PFS is not impressive, 4.9, but it's similar. Um, but I think this paper, has limitations in citation, so I think we can stop here. Next paragraph.
Yeah, so because we keep publishing our results for other cohorts, we have to at least mention 6 of our DART papers, and so that that comprises a lot of the
Ah,
references.
right, right. Well, this is just a 3 patient. Okay, next paragraph. It's okay.
This is the first line that you saw previous.
Oh, okay. Yeah, go down. Okay, these are future trials. Okay, this is Met KRAS. Okay, go down. Go down. TCGA data, okay. Okay, go down. Mm, okay. So, so TCGA is what? Correlation study. There's no therapeutic implication here, right? So it's a more like immune landscape, genomic landscape. So I would rearrange discussion so that, and this is true for everyone who writes any clinical paper, you would start with, so this is how I write discussion. Number one, summary. What is like the highlight of our study? Summary. And then you start with biology, sarcomatoid, genomic, immunologic landscape before treatment. So talk about that first, the TCGA, and then you move on to therapy. Then based on this rationale, you move on to the studies that have used different immunotherapy. So you discuss monotherapy, combo therapy. So I would rearrange treatments as well. So discuss the monotherapy, immunotherapy mono first, and then I would go to mono plus the chemo, and then I would go to durvalumab, the immunotherapy, dual immunotherapy. And then, because you discuss so many different papers, I want you to summarize. So overall, monotherapy, first line, second line is kind of in this range. If you add chemo, then it gets better. But with durvalumab, I think there is improvement in PFS or duration of response or something like that. And if after you discuss these papers, then you say, well, compared to our paper, durvalumab, like, what's the commonality? What is different? Our regimen did better than the monotherapy. Chemo combo comparable, durvalumab comparable, but duration response is the best out of all. Out of chemo immuno or the durvalumab, our duration response was the best. Toxicity wise, our paper was best. If you add chemo, there's a lot of chemo side effects. Our regimen is chemo sparing, so although chemo immuno was really good, durability, toxicity, our regimen is still the best. Something like that. You have to. I know there's a lot of things, but follow the logic and then after you discuss the commonalities and difference between our regimen and the other paper, then you move on to what is the strength, limitation, significance for the first time things, and then future studies. What are we gonna do? We're gonna look into the correlative markers, all that, and then you finish. So those are any clinical paper. Remember that summary, biology, rationale, then therapeutic evidence, and then similarities and differences between our study and prior studies, then strength, limitation, future direction. That's how you write discussion in every paper, okay? But and then the mono, combo, all that, so have that logic. Okay, to summarize, okay. Start studies has these strings, summarize, expanded trials. Okay, good, I like it. So Hasong, you became a master in writing these clinical papers.
No more.
This is your last start paper, so it's a training. It's like unless you write many of these papers, it's hard. Imagine never having written these papers and you have to write it during busy clinical rotation. It's hard. Yeah. Okay, so yeah, make those changes and I think will be a much better paper.
Stas is here, so would you like? Hey, Stas. I'll hand it over to him. Besides this, I worked on the transplant papers. Doctor will present that to you.
Okay. Stas,
Hello.
long time no see.
Yeah, I'm finally done with almost everything. I just have 2 more weeks of PM&R and that's it.
Wow, exciting.
Yeah. So kind of the big update I have is because I had time, I finally finished making the rewrites and changes to the ITH and TMB paper, and I wanted to get your thoughts on it, and then based on your feedback, I can make those changes and then I can submit it. I have put the link to the paper in the chat. Last time we spoke, you said you recommended TLCR, Translational Lung Cancer Research, and so
Yeah.
I've rewritten the paper to kind of fit their format. They're
Okay.
they're a little. It seems like they're a little bit more specific than other journals I've worked with, in terms of how they want everything structured.
Okay. But
And
they're generous in terms of word limits, I think.
yeah,
Yeah.
correct. And figure counts and table counts.
Okay.
I was surprised. And so because I had to actually go ahead.
Okay. Are you done with the paper or where are
Yep,
you?
it's all finished. I guess overviewed some big changes I had to make for the introduction and the discussion.
Uh-huh.
They kind of, I know you just talked about writing a discussion, but they have kind of a specific paragraph structure as far as the information goes. And so to kind of fill that and to fill those sections, I had to just kind of add more meat to the discussion, specifically around the rationale regarding ITH and TMB. And I found this fantastic paper about melanoma where they used TMB and ITH and discussed this idea of neoantigens coming from clones versus coming from subclones. Just because you have a high TMB that doesn't tell you anything about which clones neoantigens are coming from. And the claim they made in that paper is that, and they cited other work that I cited here, is that immune tumor surveillance and tumor infiltration is primarily driven by clonal neoantigens. And so if you have a high TMB and that TMB is derived primarily from subclones versus clones, then the neoantigens are coming from subclones, and you'll actually have less tumor infiltration. That's kind of the broad claim that they made, and I mentioned that here as some of the rationale behind why using ITH and TMB together is important, because you're able to get a better idea of that kind of clonal architecture and yeah.
Okay.
which I thought added a little bit more meat to the, and also address some of the prior concerns from the JCOPO folks who said, you know, what's the rationale? Why are you using ITH? Well, we're using it because it helps us better understand TMB. But I talk more about that in the introduction and the discussion.
Mm. Mm. I don't like the expression the highly prevalent indications. I mean, you can calculate ITH and TMB in any cancers, and dynamics may be different in all different type of cancers. So
Sure. I don't know
essential.
what that means.
Just write it more. This to me, it sounds fancy, but it's not too scientific. I would just
Okay.
follow, you know, um,
I'll just highlight it and
we,
simplify
you
it.
know, we explore the non-small cell lung cancer data from TCGA. Well, you say TCGA here, but we just say, you know, within lung cancer, we investigated the association between ITH, TMB, and its impact on survival outcome.
Okay.
Let's just write it very plainly.
Yeah.
Uh, yeah. High ITH predicts an extended PFS while in lung. High ITH. Lung, I don't know, reduced. So how did you get adjusted P?
I used false discovery rate control, I believe it's called. It just takes all the p-values for all the comparisons and adjusts them.
So so this adjusted P, this one.
Yeah.
So
That was, yeah, go ahead.
how do you adjust? Because you have low
Correct.
IH.
Yeah, so it all comes down to the number of kind of comparisons I'm doing. So if I have a subset that I'm only if I'm only using one cutoff for that subset, or like if I'm testing only one cutoff to see if that cutoff yields anything statistically significant, I won't adjust it. If I'm using more than one, then what it does is it looks at the p-values across all of your comparisons, and I think it adjusts it basically roughly multiplies the smallest adjustment by the number of comparisons. It's not exactly that, but it's roughly multiplication. It's like a less conservative Bonferroni approach. And I mentioned that in the methods,
Hmm.
but I can be a little bit more clear about it.
Okay. I wouldn't put adjusted here. I would just write P equals, and it because in the method you would explain how you got the P,
Okay.
because it for the abstract it's too much information.
Okay.
Like we're like, how did you adjust for
Sure.
it? That, yeah. So T cell enrichment. So I would actually write, what type had higher T, and then was it higher ITH or lower ITH? I would write the detail, not just say they were.
Sure. Yeah, that makes sense.
So I would say still in the background, why focus on low TMB? And why do we think we have to look at both squamous and adeno? So background has to explain, why did you look at them,
Yeah.
So I know you did in introduction, you have to say, we believe that the effect of the ITH may be different between histologies, and in TMB high and low, impact of clinical implication of ITH may be different between according to TMB level,
Okay.
something like that. And because high TMB group are likely to be the beneficiary of immunotherapy options, we decided to explore TMB low, where we traditionally think that they would benefit less from immunotherapy. So we have to have a clear answer why
Yeah.
we decided this group even in the a little bit in the background, because it
Sure.
doesn't explain here.
Yeah, no, that makes sense.
All right, so TMB heterogeneity. I know you.
So this first bit is just the background and then
Yeah.
rationale and knowledge gap. And then objectives.
Yeah, I don't know if you need subheading for introduction. I would take it out.
It's just that this is the structure they provide. This is the template that they provided.
Oh, so this journal wants you to write it? Subheading?
Yeah.
Okay.
I mean, I'm sure we can get away with not writing it, but they were like, use this template. I was like, okay, I'll use it. You know?
Hmm. Okay.
And they did the same thing for the discussion.
Hmm.
Um,
Did you make any big changes? Or is it
I should remove that. That was just for me to make sure I wrote the p-values in the correct format. The big changes were in the introduction where I wrote more about kind of the rationale knowledge gap. And then I added a results section about adeno with EGFR patients removed.
Hmm.
And then I made changes in the plots for the adjustment, for the p-value adjustment. And then I also made changes in the discussion.
Hmm.
Again, to fill out the headers that they were that they were asking for, but also to include more rationale behind using ITH and TMB together.
mm. So I would take this out. There's no clinical recommendation. I mean, it's not even a developed marker, so you don't have to say that. Just use median cutoff.
Okay.
I would take value out in all the P's, just write P. And P doesn't have to be capital. Small p is fine.
I think the only reason I did that was because of
Huh?
it was because of their statistical requirements.
Oh, journal said that? Oh,
It said to put a capital P. The
oh,
description of the p-value should be in the uppercase format.
what about value? If you don't need it,
Value wasn't specified, so I can get rid of value.
yeah,
I
yeah,
can just
there we
leave
go.
p everywhere.
Yeah. So what is the most important graph among this?
E, F, G, and H. Well, I think they're all kind of important because A, B, C, and D talk about the ITH relationship on its own, and then E, F, G, H, sorry, A, B, C, D talk about the ITH relationship on its own, and then E, F, G, H talk about ITH and TMB.
Hmm.
I mean, it is a large figure. I kind of get what you're saying.
I'm just for the readers.
Yeah,
Oh,
this is definitely the key figure, and I could split it up since now we don't have a figure requirement, but I think all 8 of them are important. Mostly the PFS is important, but I can't put a PFS Kaplan-Meier, not an OS Kaplan-Meier.
mhm. I mean, the way you organized it is so overall ITH and then among low TMB, what happens? Um,
If it's easier to read, I could switch E and F with C and D. That way the first 4 are squamous and the second 4 are adeno.
so low TMB people versus all comers, there's they look same, correct?
Um, Roughly, yeah,
Then the question is, why do we focus on low TMB?
right.
I think that's the question.
Right. It's a good question.
And then the adjusted p-values are
Higher.
yeah, looking good. And then the reviewers might ask, like, why did you hide high TMB? What happens to those group?
I have that in the supplemental.
Hmm. Is it here?
Yeah, it's at the end.
Is it this one?
It's
Oh, this is by TM.
it's the second one. Supplemental figure 2.
This one?
Yeah.
Oh, okay. So high TMB really no. Hmm. So, and then these are maybe, well, this is still decent number, right? This
Yeah.
is- yeah.
High sample sizes, except for, I mean, squamous is a little lower than adeno, but
Yeah. So you can argue that the relationship that we see is driven by the low TMB group.
yeah,
Um,
that's why we separated them.
so I would say maybe you can write according to TMB because you've done, it's not like you ignored high TMB. You have data on high TMB and say
Yeah,
overall this relationship, there's a relationship
yeah,
and it's largely driven by the low TMB group. This association that are opposite in squamous and adeno are
yeah,
largely driven. So you can kind of change that tone.
yeah. So instead of with, we do driven by, or is that too much?
Or by
Just
by
by.
by TMB. Take out low. Because you're describing both high TMB group and low TMB
Yeah,
in your paper.
it does simplify the description of the rationale why we focus on low TMB. Instead, well, we focus on TMB and we observe that low is the main driver of this.
Right. So in the paper, we write it so that we're looking at both group.
Yeah,
Now later we found out it's mainly driven by the low TMB group.
right.
Because if you had the prior title, then people are like, why are you taking out
Yeah,
low TMB? We're not taking it out. We are exploring both groups and saying that, you know, so yeah, I like this better.
me too.
Or I look out, yeah. Driver genes. So this is saying if even if we take the drive EGFR out,
Yeah,
there's a trend still.
yep,
Still high ITH is worse.
right. I mean, a reviewer might argue that, well, it's actually because you have this tail of outliers. But even if you get rid of the tail of outliers, there's still this giant gap between the curves.
Yeah,
So,
I'm okay with it. Immune cell. I wouldn't say actions needed. I would just say implication of future studies.
Okay.
I would say, so find with low TMB and change it also,
Yeah.
by not even low.
Yeah, I'll go through the whole thing and I'll
Yeah.
reword all of our, yeah. But I don't think I have to change the figures because the figures are saying
Yeah,
these
yeah.
are the low TMB patients, these are the high TMB patients.
Uh-huh.
Yeah,
Okay, cool.
great. Yeah, I think that really strengthens it, and simplifies the
Yeah,
rationale explanation. Thank you. The other thing is I'll get this done in the next few days and um I don't know if I can get it done sooner than the next lab meeting and I can send you and Haesong an email with it and then you guys can look at it one more time. Um,
yeah.
and then regarding the Tempest project, uh, it looks like they want to meet with us to discuss kind of some of our findings. I replied with my availabilities and so hopefully they'll get back to us. The other thing I wanted to show you, and I don't know if Hassan, did you show him the graph of the co-mutations?
Oh no, you show it to him.
I wanted to show you that. Basically, I was also curious to see. We were talking about driver mutations and driver mutation landscape in the I don't know cohort, and for a while now, I was thinking of ways to illustrate the co-occurrence of mutations in driver genes. And I've been struggling with that, and I think that I finally have been able to do it in a way that's what's the word I'm looking for? Kind of clear.
Mm.
Sorry, I'm just formatting my window here so that it's a little bit more. Sorry for the delay here.
That's okay.
Well, this is the best I can do here, so that's okay. All right, let me share my screen. Okay. So this is kind of the prettiest way I've come up with how to show it. Um, so okay. Whenever you're ready, Dr. Chae.
Okay.
All right, so what you're seeing here is kind of a network graph of all the the list of common driver mutations. The color of the line between them is correlated with the percent of patients with the co-occurring mutation. So for example, TP53 and KRAS, the line is yellow, which means it's close to 60% of the patients have these mutations co-occurring. And then there's an orange here and so on. The lines that have been removed are when there's only a single co-occurrence. So in the next plot, everything in green is just a single co-occurrence of these two drivers. And in this cohort, there isn't a single patient with only one driver mutation or a mutation in one driver gene. So these driver genes all happen in pairs. The question is which pair? And so at least for me, I thought this was like a better way of, or a decent way of visualizing how frequently mutations co-occur with each other.
Mm.
And so these first two are in high allele frequency,
Mm,
and the second two are in low allele frequency. So just whether it's statistically significant or not, in high allele frequency, you have a higher co-occurrence of TP53 and RET, for example, because it's yellow. While in low allele frequency, you have it's orange, which means it's less than 40%.
mm,
And then here, the opposite is true, where TP53 and BRAF in the high frequency group occur maybe 15-20% of the in 15 to 20% of the patients, while in the low heterogeneity group, they're occurring more frequently together.
mm.
And then, you know, the single occurrences.
Single occurrences? What do you mean?
So the green ones are pairs, are driver gene pairs that only occur in one patient. They only
Green
occur once.
only a co-occur once.
Only co-occur once. So in this one, I have them here, and then here I have them removed just for the visibility sake. But the only reason I did that, because in my mind, if these mutations co-occur once, there's probably not a significant survival association with them. Or at least we don't have enough data to claim any association.
Hmm.
But I don't have any grand conclusions from this, but more of a way to just visualize this type of data so that we can ask ourselves if there's any driver gene pairs that are associated with, or rather, if there are any driver gene pairs that lead to higher heterogeneity or lower heterogeneity. Because in my mind, thinking back to this paper that we were just discussing, kind of what drives heterogeneity? Is it a collection of, is it just the tumor microenvironment? Is it, I mean, it's probably both the tumor microenvironment and the changes in the cancer cells, but where do you have, what has a larger influence? A driver gene profile or a tumor microenvironment profile, or kind of, yeah, what drives the heterogeneity?
Hmm.
Anyway, that's all I wanted to show.
Um, low, I mean, you have low allele frequency, high allele frequency group. What's your cutoff? Is it median?
Yeah,
Okay.
in this case it is, which just happened to work out well for the sake of statistics.
Okay. So I'm okay with, this graph, and it could be in, I don't know, depending on what the final paper looks like,
Yeah,
final regular or supplemental. Um, I would put it in supplemental unless we find like, oh, this is a new discovery we made that
right.
they're like right now I don't see what the new discovery is.
Yeah,
Number one, because I know that Tempest gave us this mutation information, but this is all, they did not differentiate functional versus non-functional, correct? Hesong, do you know?
I don't remember.
They gave us
I
mutation.
believe no.
We don't know if these are really um meaningful mutations or not. Even like, for instance, EGFR, there's a lot of, VUSs.
Yeah,
Uh, and it's night and day if you have exon 19 deletion versus EGFR VUS. It doesn't do anything. So what this relationship really means in terms of clinical impact is unclear, really unclear. So that's number one.
right. I'm just looking at their sheet right now. I mean, you're right. They don't tell us whether they're significant mutations or functional, but I wonder if we can find, and I think Eugene Kim kind of worked on this. If we can look up a database of, pick your favorite driver gene and the known variants of known significance and be able to filter based on that.
Yeah, so that's something Sally can do, 'cause she's been
Oh,
involved.
perfect.
Yeah. Finding, at least using, one program that we know is better, or there's a combined program, right? Like Revel. So, at least categorize them into functional, non-functional, and kind of look at the functional mutation landscape alone, or
That's a good question for
combined
dentists
and
too.
functional. That might look, and the second is the whole point of the project is ITH for this Tempest project.
Yeah.
So I would draw similar in a high ITH and low ITH. And see if those co-occurrence pattern landscape is any different in ITH high group versus ITH low group. So that might be something we want to look at.
It seems, at least based on the graph that I saw, barring the functional filtration, there do seem to be, just from first glance, some differences between the frequency of co-occurrence between the groups. But I don't know if that's statistically significant,
Yeah,
like of the
so yeah,
difference. Yeah.
do that and let us know if you find something interesting. But in general, I'm supportive of these visualized approach.
Yeah.
Yeah.
It's just really hard because, especially with the co-occurring mutations, it's hard to, at least for myself, to kind of wrap my head around how do you evaluate significance of these relationships. And the visualization at the very least helps me focus on some pairs that happen more frequently. But yeah, that's it. So kind of in short, the update for that, we're going to present our results, meet with them, and see if we can get more data or have some kind of opportunities to use PyClone, or ask them now whether we can filter for functional mutation status. So,
Sounds good. Sounds good. Okay, anything else, Das?
no, that's it.
Okay. Um, yeah. I don't know. What are you doing, Stas, in the afternoon? Are you in
No,
lecture?
I'm off. I'm off basically for the month of March. I'm gonna do a little bit of traveling, see family, that sort of deal. So yeah, I have nothing going on this week.
Oh, okay, great, great. Um, okay. We have people coming over from Genomic Insight today. Is it 2:30? What time is it?
Yes, it's at 2:30. Is that meeting still happening? Because we also have that dinner meeting with them. So I wasn't sure if both of them were happening or if we're doing the dinner meeting alone.
Well, dinner is just for, networking.
Oh, I see.
2:30 is they will come and they will give us presentation, and I think we'll give them some presentation. So let's see. So let's do this. So because they are whole genome sequencing company, I think they would be interested in what we're doing with the ITH. I don't know, Stas, it's up to you. If you want to come over and meet with them, they are gonna come to our lab. So, but I feel like we could maybe Liam look into renting a room at the School of Medicine just because they have a wider screen, and I don't know if you have the cable. Um
We can probably rent a room in the library, or
yeah, the same room we use or a different, yeah, because they have a large screen. And Stats, do they have wireless connection too, or do you always need that cable?
I've usually seen people use the HDMI cable that's provided.
It's in the room.
Yeah, it's
Oh,
attached to the TV, but they
okay.
don't have adapters for, depending on your computer.
Oh,
That's something you just have to keep in mind.
right. I have an adapter, but some of the laptops, you guys carry
Yeah.
would have HDMI built into it. So, Stas, if you can come to 30, you're welcome to meet with them because I think we are planning to do MOU and forging a new collaboration with them. And they're doing the whole genome sequencing at the cost of less than whole exome sequencing. So they're like
Wow.
disruptors in the market, very low cost. And then the founders of the company, they're based in San Diego, but they're the some of the founders of the companies are from my medical school. So I got to know them, met with them multiple times. They flew in from San Diego just to meet our lab
Oh,
people.
wow.
They're going back today. So you're welcome to come and meet with them. I feel like they're gonna present to us what they're doing. But I feel like, so first of all, are you coming, can you come or not
Yeah,
to
I can
like,
come.
okay,
Yeah.
if you come, maybe prepare, like 5-minute presentation of the, the ITH project that we did with TCGA and then the Tempest. Just don't say it's Tempest, just say it's a that large database. And this is, what we're doing and what we found so far. And then I think the main key is why are we interested in ITH? So some of the tables and figures you have in your review paper, with me,
Yeah.
just make that into PowerPoint slide and say, this is why we're looking into ITH, and these are
Yeah.
our ideas. And how do you measure ITH? This is how people have done, and then we have access to raw NGS data from blood and tissue, and this is how we want to vision
Yeah.
assessing ITH from blood and tissue in the future. So we almost have access to now the Gardent, and we have Tempest, so we're at the beginning of delving into that. And then Foundation is, other companies are really interested in it, but I feel like whole genome is another level. It's,
Yeah.
if you look at heterogeneity at really different level. And so why don't you prepare like 5-minute like
Yeah.
slideshow of our what the ITH, the review paper, and the TCGA analysis and things that you've done. Uh, yeah, so I feel like that's really important. Um,
Yeah, I can do that. That should be straightforward and fairly quick.
okay.
I do have questions for them. I mean, it'd be interesting to hear if,
Of
you
course,
know,
it's time for us
yeah,
to ask them questions. So
yeah.
that
Like
would be really good.
do they do methylation? Do they do any other epigenetic sequencing alongside their whole genome studies? Are they, because with whole genome, the thing that is also not often discussed is when you use approaches like Illumina or primarily Illumina, you don't get good sequencing coverage of regions with high repeats. And so you say it's whole genome, but it's actually just big chunks of most of the chromosomes, and there's still a lot that you're not getting. So it'd be interesting to see, are they also adding in technologies that help with sequencing of highly repetitive regions. You know, how complete is their whole genome sequencing assembly? Just my own curiosity. I don't know.
Yeah, so yeah, I think there's one of the CEO is PhD, like just majored in NGS, nothing else. So
Perfect.
it'll be a good time for us to learn. And then for that meeting today, um, hmm, ITH, well, we can work with them on the ITH, but also on,
Well, the nice thing about whole genome also is it can help us delve into TMB a little bit more. Because right now TMB is like this massive average across the entire genome or all the exomes. It would be nice to see if TMB differs based on chromosomes, based on chromosome locations, if there's some association with you know, if all the neoantigens are coming from this chromosome, you get more and you get kind of a better response. Or if they're coming from this one, you get lower response because you can make the assumption that, hey, all these mutations are present, so there are more neoantigens, but which neoantigens are actually showing up on the cell surface? I don't know. But anyway, it's just my own kind of research questions.
yeah. There and then wasn't your project something based on H&E slide and the I guess the architectural heterogeneity,
Yeah,
does that translate into genomic heterogeneity?
right.
Where are you with that project?
I haven't had time to work on it. I've definitely gotten more. It's just it takes a lot of time, and I've learned kind of the basics of it. So it's definitely something I want to spend this month figuring out. The nice thing is, what I was thinking is Tempest has a lot of H&E slides for their data set. So I was thinking, and they provide the environment, they provide everything. So I was thinking um, of of using that data as a place to test some hypotheses.
Yes.
Um,
Yes, that was our initial project with Dr. Chung-Han Park. He was the first one who,
Lunit.
yeah, came and made connection with Lunit, and then he went to Jack's laboratory and studied that. And he said it's so hard to
Yeah,
measure heterogeneity with H&E slide because everything is heterogeneous, and
right.
hard. But and then how do you link that into molecular
Yeah.
heterogeneity? So
Well, I think you have to start with, at least the way I've kind of understood, you have to start with some simple questions first. For example, there's good software out there that can highlight the immune infiltrating or the tumor infiltrating lymphocytes, that can highlight stroma, and that can highlight neoplastic cells. And I think the first question you ask is, are the neoplastic cells and the tumor infiltrating lymphocytes, are they interacting with each other? Are they surrounding? Is there like a stromal cell in between them? Kind of these basic questions. And once you have those, you can start delving into, well, can we classify these neoplastic cells? Are there like buckets, like, you know, from a scale of looks a lot like the cell of origin to looks very different from the cell line of origin or something. Basically, that at least that's how I understand it. And then you chop up the H&E into tiny, tiny sections, and you kind of assign some kind of scoring system, whether you're talking about interaction between neoplastic cells and in the immune system or some other kind of metric, and then you sum it, or you come up with a way to describe it. But the other issue is the sequencing. The way that it's typically done is a molecular pathologist picks a region of some slide of some block to sequence. So what you're seeing on the H&E is not usually the same region that was sequenced. So
yeah, I think that's the biggest problem. Actually a problem.
yeah, but
Yeah,
thus
You're
Yeah.
not really, that's why blood is important. The blood will pick up everything.
Right.
So. Hmm.
But yeah, to answer your question, I'm still working on it. It's just been kind of on the back burner. Um,
Hmm. Hmm. Okay, so if you're coming, then
yeah,
just come up with a slide or a laptop or
yeah,
drive and
I'll bring my laptop.
have you and then maybe Hesong and Liam and Sally, you guys can briefly talk about maybe a couple minutes about what kind of projects you guys been doing. Um, kind of for them to understand what our labs have been working on. So um yeah. Mm, maybe I don't know. I'm thinking about maybe you guys can work on, Liam can or maybe Sally, you can make some slide about the dream. Uh, and just this is just for us to let them know what we're doing. And Haesung, you can make slides on the dream study and then the mucinous study, maybe just the abstract a table or whatever you've worked on. Maybe just prepare a couple slides showing what you've worked on in my lab. Uh, yeah, that would give us a nice presentation of what we've been doing and how we can leverage our collaboration on future projects. So maybe has on the Tempest project as well as the Dart project. Maybe focus on the thyroid translational works that we've done. Sally, we don't have to talk about the H&E slide project, but maybe just the dream project in general. And Liam, maybe you can focus on our Lunit collaboration. Because you've been doing that with Isu. Uh, yeah. Okay. All right. Anything else, Stas? Okay, good. Let's take 10-minute break and we'll resume. Haesung, are you able to read the journal club paper?
I honestly tried. It was always on my mind, but with the rank list, the sarcomatoid, the transplant, and I can't remember other things, but like I was a bit overwhelmed, so I could not do it.
Can you do it next lab meetings, any time that you could make yourself available?
So next week I'm gonna be traveling back to Korea.
What about the
I wasn't
following week?
Tuesday. Following, I'll try.
March 12th. So let's postpone that 2 weeks.
I'll try. I mean, to be very honest with you, I can't.
If you need more time, I'll give you more time.
Okay.
I think it's a really good paper. I really want to discuss it in the lab meeting. That's the bottom line. I think you can take much more time as you need, but you can try 12. If you can't do it, let us know. We'll shoot for 19th. But I understand that, the clinical papers come first, and if you don't have the bandwidth, we'll give you more time. I just want to use this lab meeting time to study good papers so we can all learn from each other. Okay. Anything else, Haesung, before we wrap your projects?
So Dr. Wu kind of took over the Musings project and he's been trying to reorganize things and he has prepared a few slides regarding that too. So maybe he can present that to the Genome Insight team and he will also show you like kind of like what direction we should be taking, what kind of analysis we should be adding, things like that.
Okay, so he'll be here in 10 minutes anyway, so yeah,
Mhm.
we'll soon. Okay, let's take a break. 원이 제일 힘들 때. Okay, I am back. So, before we get to Dr. Wu, any quick update from Brandon?
Yeah, I have a couple of quick updates.
Yeah.
So first, I'm working on the head and neck cancer portion of the spreadsheet with Dr. Wu, Uranos, and Dr. Liam. And so that's nearly finished. I just have to relook at the papers again. Dr. Wu asked me to look at the reoccurrence of each paper. So I have to look through this again. And then Uranos and I each gave a speech about like an approach to looking at systematic reviews. Uranos looked at the Amstar 2, and I looked at the grade approach. And so we each gave like a small quick presentation to Dr. Liam. I think it was like on Wednesday, I believe. And then right now, Aranos and I are using the Amstar 2 method to look at our meta-analysis papers. I specifically looked at 4 of the head and neck cancer meta-analysis papers I looked at, and I sent my results to everyone on like yesterday. And so then Dr. Wu asked me to look at a couple more meta-analysis papers. So he sent me some, so I'm going to take a look at those by the end of this week.
Okay, good, good. Right. Okay, Dr. Wangi, can you give us update?
Okay, just for a second, please. So, Sally, could you share the no, I will share. This is Google Docs, right? Mhm. So there are 4 projects are going on. So for the transplant malignancy, we sent the manuscript to Dr. Barat, and he said he will review during weekend and send us back. And until yesterday, we have not heard from him. So I sent him an email again, and he said there's no conflict of interest. So I think we can just proceed to the submissions.
Yeah.
Yeah, so I submitted it and I forwarded to your email. I think in JHLT there was your account. The email was chatemeeting@gmail. So maybe
Yeah,
you can just approve it and they will start to review.
Dr. Liam can approve it. He has the account.
Okay.
Yeah.
Okay. I will talk to Dr. Liam and I will submit it today. For the second part, about graft failure and post-transplant malignancy pan-organ analysis, we finalized the table and figures for that, and we will start the manuscript writing with Hasson, and we will finish it in maybe it will take some 2 to 3 weeks, I think. That is our goal. And as Brandon said, they are doing metastasectomy process. So because we need quality evaluation for each studies, they are doing those process. It will take, I think, about 2 more weeks. Then we will start manuscript writing, and my goal is to finish writing the paper in March. So yeah, that is the process. And for Tempest project, I received the file and the preliminary report from Hasson. And today I wanna suggest my ideas for this study. So just a
Okay.
second. So when I look at the Tempest data, they had about 655 patients of mucinous adenocarcinoma patients. So I reviewed recent papers and current literature, and I think it's more informative to go deep into the IMA only because in that abstract The non-IMA is about more than 10,000 and they are 600. The numbers are too different, but
Mm.
in IMA, the over 600 number means the biggest cohort in until now, I think. So
Mm.
it's better to go deep and review the data. So as you can see, the left one is IMA, which has cytoplasmic mucins, and the right side is non-mucinous adenocarcinoma. So I
Mm.
want to just briefly review the current literature and suggest my ideas.
Mm.
So this is typical, IMA presentation. So sometimes it looks like very solitary. Just a second. And sometimes the most new characteristic of IMA is it looks like pneumonia. So many people confuse, in this patient, the left side is pneumonia and the right side is cancer, IMA. So it's very hard to diagnose because of radiologic figures. So this paper is very interesting paper. Here they analyze the radiology characteristic and they categorize into 4. So
Mm.
A is solitary region with GGO around.
Yes.
B is only solid tumor, a solitary region. C, they call it as crazy paving appearance. It looks like it is the term by themselves, so they are making something. So it looks like pneumonia or something like that. So but they have two different. The D, if you see D, it looks like definitely pneumonia appearance, but this is another difference. So they separated in solitary type AB and the pneumonic type CD, and they compare the outcome. So
Wow. They're
it was,
creative. Yeah.
I think it's very interesting topic, and we can think about how to use this data because their number is, 38 and 32, just 670 patients. So they
Oh.
compared it with in radiologic features, and this is their analysis. They said when they compared the gene expressions, solitary and pneumonic type, there was no difference in EGFR, KRAS, BRAF, and other things.
Mm.
However, and they said you can see these 4 types of IMA, right? And the size was different. SUV max is different. But interestingly, the mucin MUC1, MUC6 expression pattern was different.
Oh.
So they suggested this can be some kind of suggestion. So this is their idea. It is quite interesting. And so they compared the mucin expression pattern and suggested as prognostic factor.
Mm.
And another issue is, when we see IMA patient, there are many multifocal and bilateral involvement. So many people concerned, is it because of late diagnosis? Because clinicians thought that this is of pneumonia, or this is because of stars. So, and also another question is, if they are spatially separated, does this mean that their clonality is different? So there are 2 papers about this issue. One is from Korea. I know that Dr. Chae and Dr. Shin very well, and they they used about 8
What
patients.
group is this?
They are from Severance, and this is also combined with Samsung Seoul Hospital. So
Oh, okay.
this is very unique because they analyzed 8 patients with multifocal vision and they made a phylogenetic tree. So based on the
Oh, okay. This is what Dr. Barat is doing in his lab. All
oh
the dream,
really?
all the dream patient,
Mm-hmm.
we have whole long samples of each different IMA site. So
Okay.
we're sampling all that site bilaterally
Okay.
and making
Okay.
the phylogenetic tree.
Oh.
So
So,
yeah, from all the dream patient. So that's
mm,
our next, project. But
okay.
anyway, someone has done it before, I'm sure. And then Charlie Swanton, and I we as a lab did some journal club with them. Haesung, did you do Charlie Swanton's one? Did you do one paper, journal club, or do you know
I
who did? Was it
believe so.
Myungwoo? Do you know who did it?
It was me, I believe so.
Yeah, yeah, yeah.
Probably
But Haesung
me.
did at least. I mean, there were in one science that uh issue. It was all Charlie Swanton's paper, and he's like my role model. Charlie Swanton.
Mm,
And
okay.
he does a lot of this phylogenetic
Okay.
analysis. But so I met him several times. We
Mm,
we actually had lunch here at Northwestern restaurant here. But
mm,
he said he had some IMA and he did some analysis with his paper. It's published in Science. So
mm,
you can look into that paper. He said it's
okay.
in the supplemental figure. So he has
Okay.
recent phylogenetic analysis with his autopsy samples, and
Mm-hmm.
it's been recently published there in Science. But then he noticed that there are just bilateral lung and no other spread patients, and he was interested in their phylogenetic tree. And yeah, he said he had a supplemental information about those patients, but we can look at them together later. But just
Okay.
for you, yeah. Okay,
Yeah.
let
So
me know what you found.
also here they analyze the mutational signature. I don't quite understand what signature means. It seems like some kind of cascade pathways.
Yeah,
So
I
they
think it's like Apple back smoking
mm,
signature. There's, with the ATCGA, what's, there's a pattern they define. That's probably what it is.
so they try to use this one to apply the prognosis.
Hmm.
And there is another study about this is from MS Memorial Sloan MSKCC. They analyzed 24 patients in spatially separated lung region, and they also they used DNA-based NGS and RNA-based NGS, and they defined clonal as if two or more shared somatic mutations, and they said it's clonal. But interestingly, nearly all separate pulmonary IMA represented intrapulmonary spread arising from single tumor, single clonality. So here they analyzed 24 patients. So they followed up, and they checked the mutations. And other than one, this number 8 patient, they all have same kind of clonality. And interestingly,
Wait, I don't understand. Go back. These are all
yeah.
patient
Yeah, this is number 1 patient, time 1 and time 2, tumor 1 and tumor 2. So separate the reasons.
separate
So
lesion. Okay. But
yeah,
they
yeah.
all
So
have shared
shared
pathogenic
other
mutation.
than other than one patient.
Other than one patient, everybody has
Number
shared.
8. Yeah. The interesting thing is there were 4 patients who had a lesion, and after 8 or 9 years, another lesion came out. But the
Oh,
two regions had same clonality. So it's very interesting. So this kind of approach, maybe we have about 600 patients. Maybe there would be some who did biopsy again. So maybe we can compare them. So this is some idea that I got from them. Also for in genetics, these are kind of pie chart for IMA, but
wait,
the
can you
total
go
number,
back to the clonality, the tree, phylogenetic tree?
phylogenetic tree,
So from this, what did they find?
they said 2 patients had different, but
Clone?
other 5 patients have same origin. So
Two are different, and other ones have shared.
shared, yeah.
Mm, multifocal. Okay.
But this is multifocal, and it is also in the same time, not in the different times. So, but
Oh, this is
in
same time. Okay.
yeah, but in this study, they included the recurrent cases and they compared the genetics, and they said even though there are two, time lap about 8 and 10 years. However, they have same clonality. So it was so interesting.
Okay.
Yeah. So this is from about 400 patients. Actually, they included 5 published data and made this kind of chart. And they said that most common type is KRAS mutation. However, in the codon 12, the replacement of amino acid is different than non-mucinous adenocarcinoma. So in non-mucinous, G12C is the most common, but as you can see here, G12D and G12V is the most common in mucinous adenocarcinoma. So when I looked at this one, and I think because we have more number of patients, I'm not sure whether we can draw this pie chart again, because this pie chart is from 5 different studies. So there can be some problem of combining all the data. So if Tempus can do that, maybe we can make an updated or new version of pie chart.
So A is A IMA, B is non-IMA.
No. All are IMA. But here, these are all mutation. Here, they are only analyze KRAS mutation.
Oh, KRAS.
Yeah, only KRAS. So
So
they said,
I think with Tempest data we can make this same type of
okay.
table. Yeah.
So they said G12V and G12D is most common in colorectal and pancreatic cancer. So yeah, this was so another interesting part.
Yeah, I think it's really interesting that this is why that usually the IMA is less responsive to
Mm.
the conventional therapies.
Yeah.
Yeah. Okay.
Also, they also in another study they compared tumor the difference between IMA, non-IMA, and they said the KRAS, EGFR, TP53 is very different, and also mutational count is in mucinous is much lower than non-mucinous. So this is another figure from that I didn't understand quite well what they are saying. So yeah, I will review and ask you again. And also we have some data about mucinous and non-mucinous. It is also same in tumor mutation burden. So we can give some update about these results. And they said after KRAS, the second common is NRG1 fusion. So, so this is about NRG1 fusion is about 6% here. So they talked about it. And another one is third most common is the RBB2 mutation. I don't know what these mutation means in clinical practice. So
it means that you can use HER2 targeted therapy, and the ones we use now is called NHER2. Works really well. I think it's going to be frontline. Currently it's second line, but it works really well. The toxicity profile is really good. So NRG1 and HER2 mutations are really good target, but it's still
okay.
yeah, not. I mean, it's more
So
prevalent than non-mucinous, but still it's not majority. So
yeah, okay. So they also mentioned some non-driver mutations. So yeah, this is something that they said this is a common non-driver mutation published before, but we need to update. For the treatment, they said PD-L1 expression is very rare. They reported 1 to 10%, but in our data, about 29 or 25% were PD-L1 positive. I'm not sure what was the cutoff, maybe above than 1%, but in many papers, they said immunocheckpoint inhibitor targeting PD-L1 might not be beneficial to IMA, but some suggested B7H3 might be better target. So, and there are not many reports about the PD-L1 positive IMA's response about immune checkpoint inhibitors. So it can be our topic. So another one is we have a data about T cell infiltration, right? So in terms of tumor microenvironment, this is the only paper about invasive mucinous adenocarcinoma.
Oh, okay.
Yeah, so it means we have more updated and more number of patients. They compared CD8 negative and positive, and they said CD8 positive is that survival is not good. They couldn't describe why it is that, because it seems, yeah, because if it is positive, it seems much better in other cancers, but they said they don't know, but it is the maybe some interaction between other things can make problem different. So this is another question mark, but we can do that. And in terms of clinical outcome, this is this is paper that I published. So this is only compared about stage 1 or early stage, and I graded the non-IMA into 3 grades. So grade 1, grade 2, grade 3, and IMA was around grade 2. So this is from some in surgical
Wait, wait.
database. This,
Wait,
yeah.
Can you tell me about the title? So
the-
IMA
so
compared with non-IMA? Is that
yeah. So by the purple is IMA, and other 3 are non-IMA, but in 2020, pathology group suggested a new grading system for non-IMA. So they divided into grade 1, which is lipidic dominant. Grade 2 is acini and papillary dominant. Grade 3 is something includes more than 20% of a high grade proportion, such as micropapillary, solid. So if they have anything over than 20% of high grade, they grade it to grade 3. So we compared it with IMA, and IMA was similar to grade 2, less than lapidic, but better than high grade non-mucinous adenocarcinoma. So this kind of things reported in surgical database because they only include such staging 1 or 2, but this is quite interesting paper, stage 4 invasive mucinous adenocarcinoma. The number was not that much. I remember around 60 or 70. And they compared all IMA stage 4 and all non-IMA, and the overall survival was not different. And they compared non-TKI chemo. Also, there was no difference between that in terms of, and if you see look at the chart D, this is interesting. IMA untreated was better than non-IMA untreated. So still, in terms of prognosis for stage 4 IMA, there is not much data.
But this is saying IMA untreated is better?
Than non-IMA untreated. So they said IMA has some different courses. So
Oh, okay. So I guess small sample size, but still provocative that IMA has, is all more on the favorable side.
yeah, I
Yeah,
think so.
that's really interesting. Can you go back to your study? So is this just a surgical case? Anybody treat stage 4?
Just surgical cases, only resectable cases. So stage 2 on early stage, yeah. There was some neoadjuvant something, but majorities were stage 1 and 2. Oh, yeah.
Okay.
So from these background, I suggested some idea I want to discuss. So I think we can redraw this pie chart, and we can see how gene alterations in IMA, and also we can compare it with non-IMA, but it's better to focusing on, I think, over 600 patients over IMA. And also we can compare if, the prognosis between the KRAS and NRG1, if the mutation different, if they have different clinical outcome, or they have different demographics or staging, I don't know. So we can compare it. So another one is we can divide into resectable and non-resectable, or stage 1 or stage 4, and compare is there any difference in gene alteration in IMA. then we can suggest if some specific gene alteration is evident, then maybe can be a risk factor for distant math or something. So this in about gene alteration, I think we can do this project. Also,
Can you
I
go back?
wanna,
Can you go back?
yeah,
So KRAS, yeah, and then see if there's any difference. I don't know if we have enough sample, but KRAS C versus other, you can look at
Mhm.
that as well. NRG1
Okay, okay.
fusion, um,
Might be too small.
might be too small, but still confirming that it's more than non-mucinous, I think. And then I know they have very limited transcriptomic, but you
Mhm.
can ask for the MUC1 and 6 expression level.
Okay.
Because it's a big data, they maybe at least 20-30% would have the, because what happened to my patients is that by default, they will do the whole RNA sequencing, but then
Mhm.
that's the least of their priority. So they will use samples out for PD-L1 and NGS, and if there's sample left, then they would run RNA sequencing. So we, in theory, have gene expression level for every gene, but because of insufficient tissue, maybe it's 20-30%. And what do you think, Haesung? That the fact
Mm,
they're if you
RNA
looked
sequencing
into it,
data is only for 20%?
that's what I feel. What do you think, Haesung?
Ah,
Because we asked for that data, but they didn't.
They didn't give us the exact stats.
Uh-huh.
Well, they use the RNA-seq data to analyze the immune cell infiltration patterns.
Yes.
So maybe if we look at the sample size for that, we can kind of
Yeah.
infer.
Yeah. So yeah, it's they, I think they use maybe X cell or similar program to calculate immune cell infiltration, and even HLA typing, everything based on the RNA-seq. So they do have that data, not for everybody, but if we can ask for some of the signature or some set of the genes you want the expression level of, you can ask them for that expression level, the raw data too. And definitely include the MUC. Maybe you want to discover something new. Ask for MUC 1, 2, 3, 4, 5, 6. Oh yeah. See what maybe for their paper, because it's always, it could be random error too, but there was clear pattern. So we want to replicate that finding and maybe do some more discovery. Maybe it's a set of study more about the MUC mucin. And sometimes I don't know. I went to AACR and ASCO workshop at Vail, Colorado,
Mhm.
and my small group actually, yeah, it kind of gives me goosebumps. But we were talking about mucin, how mucin is so slimy that it protects cancer cell from being attacked by immune cells. It's a slimy monster. And then how mucinous pancreatic cancer, colorectal, like all these mucinous ones are the ones who do not respond to immune therapies or cancer vaccines or anything. So how does mucin, the biology of it, affect PD-L1, CTLA4, CD8 cells? We don't know, but at least, in the mucinous setting, we can find something unique. Does mucin production have inverse relationship with PD-L1 or CTLA4 or MH class 1, 2, or JAK-STAT pathway, how, or KRAS, is there any relationship with MUC and then many other factors? We can look into that now that we have, largest of mucin. It's not single institution, like Tempus is a worldwide database. So I think we can ask all those questions. And then distant mets. So we have that data. Haesung, remind me what clinical data did they give? Did they give us an updated data on OS and
No, not yet.
metastasis?
No, yeah, it's still cooking.
Okay, but they are looking into it. So yeah, so the
Yeah.
clinical factors, yeah. Uh I don't know. Would they have any information on whether this is a bilateral or unilateral cancer? Maybe
I
it's
think they don't have radiologic factor, I think.
Right. You know, the other thing we can work with is because just like Lunit, they have their own AI pathomics division. And the pathologist working there used to be my mentee, Tim Baxter, Tim Taxter, Tim can't remember. Liam might know. Tim Taxter. And he was our pathologist fellow here. And he joined Tempest after he finished his pathology training here at Northwestern. So we have a good contact there. I wrote a textbook with him too. Anyway,
You mean he has data about IMA?
yeah, no, he has
Mm.
H&E slide and he has access to the the till calculation by H&E slide, or like any pathomics H&E slide based analysis, because Tempest also scans all their H&E slide into their database. So that's a very valuable information. So we can use that to analyze the IMAs. So think about that as well. We can,
Okay.
yeah, we've developed the TLS algorithm with Lunit, but of course I don't think we need to get Lunit involved, but Tempest, think about H&E based analysis and the correlations you can do, starting with TIL, because you can measure TIL. Yeah. Okay.
So, and if they have multiple biopsy tissue, maybe I want to do the phylogenetic trees again. So yeah, and that's
Yeah,
compared
that
to
but that's
nationality.
their commercial. So they met. So Haesung, you can ask them that, do we have a same patient with different time point tissue biopsy? Because that's rare. But I do it all the time in my clinic. So some of the investigators would be doing it. So we can kind of, yeah, say, and then what is the time interval between the two, and where did the sample come from? Is it one from lung, or is it one, from bone or adrenal gland, or maybe that will help us draw phylogenetic tree.
Yeah.
And then ask Tempest, because these mutational signatures analysis is very standard, AppleVac and all that. So they have done it with their prior papers, I know. So ask them for each patient, can you generate a signature? Can you guys generate, because they have their own bioinformatics team. Can you generate mutation signature for our patients? You don't have to do them. They can give you just the output. Signature A, B,
Okay.
C. The proportion in each patient, each sample would be good enough for you to, do the analysis. They can
Okay.
do that for you.
Okay.
Okay?
So also in our data, there was, biopsy sites about how they were diagnosed, and there were a lot of things from other than lung or thoracic cavities. So because IMA is very similar to kind of pancreatic or colorectal, so maybe if they are found in the GI organ, maybe the origin can be different. So that is, I don't know, another questions. And for the
Wait,
treatment,
I don't,
there-
I have to, the,
Yeah.
so recurrent and non-recurrent,
yeah.
is this same as? So are you like
Yeah, I think same
surgery
is,
cured and then there's something recur? Yeah. So
yeah.
that's hard to find because these are again commercial data. And it's hard, you have to read all the, you know, the chart to figure out if this is a recurrent one or this is, you know, simultaneous one. So I would say this is going to be really difficult, unless you read the chart. But I like this GI organ. This is why I
Yeah.
told Tarek to work on,
Mhm.
you know, FOLFIRINOX, Zilox, FOLFIRI, all these GI-based regimen chemo.
Mhm.
Would it gonna work better for mucinous lung cancer? Because it looks so much in GI, KRAS G-top-C is very small. It's all D and V. So even like a lot of genomic landscape looking like GI here. To a point where we are always confused. Is this pancreas cancer that came too long? Because if you run it, the mucin and all that looks like pancreatic cancer. But
So
we've
do
been
you have
treating.
data
Uh-huh.
about treating them based on GI regimen?
No, no one has been bold and courageous enough to, you know, do that. So I told Tarek to make a clinical trial for that.
Okay, okay.
But I think it makes sense to like mucinous pancreas, mucinous colon. There's TCGA data, so we could compare at least using the TCGA data, their expression and their genomic alteration and kind of put that side by side because I don't think Tempus will give us their pancreas and colon data. But we can, I mean, it wouldn't hurt to ask. They said, well, we limit to 500. I hate that 500 rule. Like, why do you have that rule? You guys have access to every patient. So, you know, it's just, you know, barrier to scientific advancement, but we could ask like if we want to show similarity with pancreas and colon mucinous, would you guys be open to exploring that and make comparisons for us? You don't have to give us the data, but you guys can kind of
Analyze.
analyze it and compare it for the sake of comparison only. It's not a separate project, but we're asking for your, you know, support. So
Okay.
maybe they'll be open to it. Yeah, but if we show that they look very similar, genomic transcriptomic signature, then it gives us more buy-in into the therapeutics has to be similar. You know what the other one is? Ovarian cancer and lung cancer transcriptomic profile are very similar. So that's why carbotaxol works so well in ovarian cancer, just like lung cancer, carbotaxol. It's like the backbone of both. So any pharmaceutical company developing lung cancer drug, they go to ovarian cancer. That's why I have so many gynecologic patients in DTI. That's the only reason why lung and gyne cancer, because same chemos work. So I feel like I'm a proponent of mucinous is GI cancer. Yeah, but it's so rare that investigators haven't been looking into it, you know. But I feel like the best advancement comes with therapeutic application. Like, well, you can look at the phylogenetic tree, PD-L1 level, but if you don't design a clinical trial based on what you found, patient's not going to drive any benefit.
Mhm.
So every scientific finding you discover, for all of you here, the message for you guys is design a therapeutic trial. And that could be biomarker based, ITH based, PMB based, whatever, NRG1 based, KRAS based. It has to have solid foundation, and it could, and the more biomarker based it is, your trial is likely going to succeed. A lot of good drugs have been developed and they didn't find the right population, run the trial, there are few durable responders. And some are cured. But the trial always fails because they enroll, non-relevant patients, too many. They want to enroll fast, and then they don't meet their primary endpoint, and FDA kills the drug. Okay, so it's always better to know your right population first, and then design biomarker-based trial, and that's the last trial that Haesung and I have been working on. So, yeah, I think in mucinous, sometimes it's hard to say, what's the right target. Maybe it's the ratio of MUC 6:1. I don't know. They want something, a biomarker that is measurable. And because mucin is sometimes mucinous features mixed with papillary, and like then we don't know. Like, is this how much mucin is maybe so I'm a proponent of we need something like PDL1. If we want to target mucin and give GI regimen or give transplant, if it, we want to come up with a very simple marker. Is it IHC of MUC 1 and 6 or 1 and 5? But what is the marker that we want to use other than, of course, pathologist has to say mucinous, but is there a marker with numbers that shows the spectrum? Maybe, yeah, Dr. Wongi can continue to read on that as well. Anyway, I like the GI origin thought. Okay.
Yeah, sorry for writing in Korean.
Oh, yeah, that's okay.
So for treatment, we don't know the data about immunotherapy and target therapy outcomes. So if they will update you about data and clinical outcome, and we will look at and we will more finalize the questions. And about T cell infiltration, there is, as I mentioned, there's no information about tumor environment. So we can analyze our data and suggest is there any difference in prognostic factors. So finally, maybe as you mentioned, if we can make some kind of biomarker or grading system for IMA, it will be so nice. So, and there are many questions. Maybe they don't have radiology data, and I don't know much about RNA NGS, and I'm still studying about it. So, yeah, I will need to discuss with
So
Tempus.
it wouldn't hurt to ask. Tempest spot, right? I don't know. Haesung, were you at the Tempest meeting? They showed us about their radiomics. I think Sally and Liam was there too.
Yeah, I wasn't able to make it because of another
Yeah,
business
they
meeting.
bought a radiomics company, and they're doing pretty good job. But I don't know for this mucinous cohort whether they have, archived the radio. So ask them, and if it's just even if it's 20-30%, see if we can have access to them, because we can do auto segmentation, we can do a lot of different things. And now, Dr. Wongi, let's say if we have access to their, let's say, you can kind of mark it solid, this is pneumonia type, and you can kind of validate what they found, because that paper has never been validated. So you could do that too. Yeah. And I think this is a great idea for our own dream patient, you can categorize them into what lesions are, what type, and for even describing our dream patient, you can use that, because not all the mucinous are the same. And in
So
my
you
question,
mean for the dream patient means before they get transplant?
right,
Because how they were treated, something like that, right?
yeah, how they were treated, but what was the pattern of their spread? Because we can backtrack those dream
Okay.
patients and say they sometimes start with 1 GGO, and the next scan second GGO, and third GGO on the other side. And they get thicker and thicker. So I feel like describing how they spread the dynamics radiographically over time for the dream patients in any nice visual way would be really good. So here's the thought, because connecting this to the dream project, one idea is, well, so this is what I told Hasong about the Tempest, because what I want to know is in this IMA, who's bilateral lung limited, doesn't have to be bilateral, even like who's lung limited, who is not lung limited. And it might be just simply the matter of time. It hasn't spent, it has spread enough for this. But I've had two mucinous groups. One, I've had 30-year-old Korean lady who's a photographer working in New York. She came to me with bone pain, widespread adrenal gland, bone, liver, everywhere. It was typical mucinous, invasive mucinous. And every therapy I gave her did not work. She passed away in like 6 months. 30-year-old, okay, never smoker, Korean lady. I've had 70-year-old who come to me, and it started like pneumonia, and it was just keep spreading. And she never was symptomatic at all, but it just kept spreading. And we used different chemo, it never worked, but only lenvatinib worked. Albert Curie, only lenvatinib worked. It's a dirty, promiscuous PKI, we call it. It touches on every pathway, but mainly VGF. So I feel like, but what my point is, there's slow growing, lung limited STAS, called STAS, air spread through airway space. And that IMA, I would call it maybe, well, IMA is basically STAS, maybe STAS only type, but there's IMA that goes to lymphatics and bloodstream early on, and they are really aggressive. I've experienced two types of IMA. So when we're dealing with two different subtypes and categorize it one, it's hard to make any discovery. Do you understand? Because it's all confusing. It depends on how much of one subtype is in one cohort. So what I'm trying to tell you is, yes, what Hasong showed us last time with her abstract was that, yes, they tend to be more local, but there are subgroups that are distant spread and seems aggressive. And there's no way to tell, because this is one snapshot. Like, maybe aggressive one got caught early that we're calling it lung limited. So we don't know that for sure. But most people come and get Tempest when they are symptomatic, sees medical oncologists, because surgeons don't send to Tempest usually. It's the medical oncologist who sees them at stage 4, and there's usually symptomatic. So that's why I asked Hassan to ask Tempest to say, what's lung limited, what it has the distant mets. If it's distant mets, where did it go to, and what are their overall survival differences? But then if you are able to separate type A and B, A is STAS only, B is STAS and lymphatics and bloodstream, then you can look into the transcriptomic, genomic, MUC pattern difference, radiologic difference. Would I imagine that the type B would have more solid than pneumonic type. So can you show that the solid type are the ones with more early distance spread? I don't think anyone has shown that. And I think we have many good questions to explore in this mucinous type.
Yes.
But yeah, my main clinical focus is what are the biological differences between two different IMA types? And
you mean
and
long
that's
limited and others?
right, long limited and
Okay.
others, because that's the critical question for DREAM. Like we can't transplant all IMA, and then long limited at this point doesn't mean that they are gonna stay long limited forever. But if we show that biology wise, this is the biology that is usually does not go outside based on these markers, and this is our data, people are not gonna argue about it. Well, because they know all IMAs are different, but if you say within IME, these markers are related to stats only, these markers are related to distant met, then it's a better for patient selection for DREAM. Yeah. Yesterday I went with Dr. Suna, and we found a case, double lung transplant, and well, I had two cases. No history of cancer, got lung transplant, explant has cancer in it. So I said, oh, this is new discovery of cancer. Let's do a PET scan. And now I find first PET scan 2 months after lung transplant, and we have adrenal gland, bone, large metastasis disease. So I felt so frustrated about the case. But how do you best select patient based on their biology, biomarkers? So I really think this is a gold mine. Yeah. Okay, so let's move on.
So yeah, this is the end of the slide. So for this topic,
Yeah.
so
Look at HLA. Maybe it's related to unique HLA subtype. Tempus has a HLA. Tempus has all immune cell infiltration. Tempus also has immune score. Tempus calculates. So maybe Liam, you can, or Haesung, you can show Dr. Wongi, go to the Tempus website, go to each patient's example, and go to the research only section and click on all the immune variables. It will show you everything they calculate for you for your research. There's immune score, immune activity score or something. There's so many things that they uh have for you in their website. They don't necessarily put that in the PDF file where we scan it in the Epic, because those are research only, but they give you all that information. This is different from other companies. That's why I solely work with Tempus, because they give you access to the raw data and give you all the research parameters. And then when you write paper with Tempus, make sure all these mutations are germline validated, because this is the only company that does for germline. The other ones use just the SNP filter to say this is the mutation, so it may not be correct. So they give you germline too, germline. But anyway, because you asked there, so anything else?
so for world long, we will focus on IMA, right? We don't compare IMA and lipidic. So we will go deep more in IMA, right?
Yeah, let's do
Okay.
IMA, finish IMA
Yeah.
first, and then if we have bandwidth, let's look at lipidic later.
Okay. 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 Yeah. I will talk with them and I will give you feedback.
Very good. Okay. Sally, do you have any update?
A few, but do we have time?
Yeah, can you do it in 5 minutes, maybe?
Okay. Okay, then in that case, I will quickly. So this is the update about EDW. We have around 1,336 patients left. So we're going in a steady pace. So hopefully we'll try to get it done in a couple weeks. And then so VC projects, so Eugene, revision is submitted. Preprint will be submitted by Eugene, and then VC2, it's the same as last week. Grace Lee will be working on it near future, and then VC3 project. So they're still working on figures, so they said they'll submit soon, but they're wondering which journal they should submit the manuscript.
Oh. Well, Helion took their paper, right? So try Helion.
Oh yeah, okay. I'll let them know. And then translation, VC4 project, we're still in the process of revision. And I actually wanted to show you their comments, but I can bring it to you later when we have time.
Yeah. Bring it to later.
Okay. Okay. I think that's, and then I've been just working on like the consenting dream patients, and then the red cap, we're trying to change some variables around to accommodate for many different instances for each cohort. Because I know for cohort C, we're trying to split into 3 different categories as well. So we're still in the process of trying to grasp the big picture and also think about all the variables that we need to collect for each cohort. So that's something that Dr. Liam and I are still working on. So we'll bring it to you once we kind of have like an idea of which variables to add. But we're thinking of probably making different red cap for each cohort, because you might need to, you might need to collect different information for each cohort.
Yeah, that sounds reasonable to me.
Okay.
Okay, very good.
Okay, then I'll bring the revision comments later to you.
Okay,
Other than that, that's it.
okay, good. And Dr. Jung, I know you're coming soon. Are you when are you coming to lab?
This Friday,
No, when are you arriving?
2 days ago.
2 days.
2 days ago, I came to Chicago, and then this Friday, I'll be in the lab.
Where are you now?
Gold Coast? Yeah, I found the good apartment in here, Southwest. Yeah.
Oh, so you're settling down. You're in Chicago time.
Yeah.
Okay. Okay, I didn't know where you were, so. Okay, so I'll see you on Friday, but Liam and Sally can still help you get settled down. You ask them any questions if you have, okay?
Okay, thank you.
Okay, very good. Okay, so Liam and Sally, bring all your questions in the afternoon. We will wrap up here and convene here for the graduation. Okay. I will see you guys. Stas, so I'll see you at 2:30. Just come here, over here. I think we'll just walk over together
Okay.
as a team.
How do
Actually, quick
how do
update.
yeah, go ahead. Sorry, go ahead.
Yeah, so I requested to book a room in the library. Unfortunately, they have PBL from 1 to 3, so they're telling me we can either book from 3 p.m. onwards, or we might have to find a different venue.
Oh, okay. Just book it from 3. I know the Simpson building, Sally, the Simpson building has some rooms with a monitor too. Right?
Yeah, yes.
Can we book those rooms, or are they part of library, or?
I don't think they're part of library.
So can you find any room there?
Okay, I can look into it after
Yeah,
that meeting.
Yeah, try that from 2:30. If not, we'll just start at 3. We'll just chat with them, walk and kind of give them tour or whatever. But Stas, you can still come to our lab at 2:30 and figure out what we can do.
That sounds good. How do I get into the building? Because I remember last time I think someone met me downstairs.
This building?
Yeah.
Well, during working hours, there's
They
no.
let you in? Okay.
They let
As
you in,
long as I
yeah.
have my ID. Cool.
Yeah.
All right. What floor? Remind me.
Oh, 10.
10.
10:06. 10:06.
10:06. Got it.
Okay. All right. I'll see you guys then.
Mm.
Bye.